This is going to be your Chapter 1, Nature of Science video lecture. This comes from Section 1.5 in your Principles of Life textbook. Biology is the scientific study of living things. Now, living things are all the diverse organisms descended from a single-celled ancestor or a single common ancestor. Key concepts that 1.5 are going to cover is science is based on quantifiable observations and experiments. So like we said, biology is the study of life. Biology consists of three domains: bacteria, archaea, and eukarya. And then you have the kingdoms that follow those. We are a very diverse universe with many different types of organisms that exist. Now in AP Biology, there are four main big ideas. The first big idea, one, is the process of evolution drives the diversity and unity of life. So evolution is the change in the genetic makeup with natural selection as its driving force. It is the core theme of biology. Big idea two is that biological systems utilize energy and molecular building blocks to grow, to reproduce, and to maintain dynamic homeostasis. So if you know life is an open system. It needs input of energy to grow and reproduce. And there's also going to be an output of energy. Energy flows through the system, it goes in and it comes out. Life needs that constant input to survive. One way that they gain energy and release energy is through cellular respiration and photosynthesis. Big idea three is that living systems store, retrieve, transmit, and respond to information essential to life processes. Basically, all living things are made up of the same thing, DNA. The same basic structure of DNA, the same molecules that make up DNA exist from organism to organism, any living organism. And this DNA is what carries the biological information from one generation to the next and is essential for all life to exist. Big idea four is that biological systems interact, and these systems and their interactions possess complex properties. So if we look at living systems and organisms, you start out with your smallest molecule. We're going to start out at the cells level. It goes smaller than that, but we're going to start at cells since we're biology. Then cells make up tissues, a bunch of tissues make up organs, a bunch of organs make up an organ system, a bunch of organ systems make up an organism, and a bunch of organisms make up populations, which make up communities, which make up ecosystems, which make up the biosphere. As you go through each level of these, the complexity will increase, okay? And their interactions are key to the survival of every system around them. What is science? So in Latin, science means to know. Inquiry is at the heart of science. Science describes how the world works, and in order to know how the world works, we have to constantly ask questions. Inquiry is that search for information and explanation. There are two main processes of science. You can have discovery science, and then you have hypothesis-based science. Discovery science is inductive reasoning, and hypothesis-based science is deductive reasoning. And I'm going to go into those more in just a minute. Now, discovery science is how you describe nature through observation and analysis of data. Okay, basically, it's a methodology which emphasizes the analysis of large volumes of data with the goal of finding new patterns. It uses data or recorded observations. There are two types of data we can look at: qualitative, which is a non-numerical data, and quantitative, which is numerical data. Now, discovery science uses, again, inductive reasonings. This is where we derive generalizations based on specific observations. It looks for a pattern or trend and then generalizes information from that pattern or trend that it observes. Khan Academy has a really good video on explaining the difference between inductive and deductive reasoning if you'd like to go and visit that. Now, this inductive logic is what leads to tentative explanations called hypotheses, which would then lead to our hypothesis-based science. That is how we use deductive logic to make predictions. And we use experiments to design, experiments are designed to test these predictions. Now, a hypothesis is what makes predictions that can be tested by recording more observations or experiments. If you can't test your hypothesis, then it's not a hypothesis. It has to be testable, and results can either support or refute the hypothesis. Now, a lot of people write, if I do this, then this will happen because blah, blah, blah. That's a simple format of how to write a hypothesis. It doesn't necessarily have to be in that format, but there has to be a cause and effect statement of some sort. You can't just say, my hypothesis is correct. You have to tell me what it is, why it is, and what's going to happen because of that. Observation is enhanced by technology. Microscopes, imaging, genome sequencing, and satellites have all improved our ability to make phenomenal hypotheses and then develop phenomenal experiments based on that. So those observations, however, have to be quantified by measurements and mathematical and statistical calculations. Now, there are certain methods that we go through once we made our hypothesis. Again, the first thing that has to be done is you have to make an observation. Then you have to ask a question. Why is this? How is this? Where is this? Then we're going to form a hypothesis to answer that question. We're going to make a prediction to see what else will be true if our hypothesis is correct. From that, we are then going to go and design, conduct an experiment that uses quantifiable data, that's numerical data, to test your prediction. Now, once we get our numerical data, then we're going to apply a statistical test to it to evaluate to see if the results are significant or not. If the results are significant, then that means you've supported your hypothesis, and your experiment can then be repeated and results can be verified by other researchers over and over. And then we can go back and ask a new question and start again. Now, if our statistical results do not support our hypothesis, we need to go back, look at our experiment and say, were there any uncontrolled variables that we did not account for? Were there any variables we did not account for? If there are, then we need to go back, control for those variables, and then revise our hypothesis and go through the experiment one more time. Now, there are two main types of experiments that we're going to talk about: controlled experiments and comparative experiments. Controlled experiments manipulate the variable that is predicted to cause differences between groups. There's an independent variable and a dependent variable. The independent variable is the thing that you're changing from experimental group to experimental group. The dependent variable is the response that happens that we are measuring between those groups. Now, comparative experiments look for differences between samples or groups. They take large samples or groups and look for differences or generalizations. These variables cannot be controlled, and data are gathered from different sample groups and then compared from there. Because of that, it's important to use the statistical method to validate that data, to determine if the difference between the groups are significant. Statistical tests start with a null hypothesis. That's just a hypothesis that states that no difference exists between these groups. Then statistical methods eliminate the possibility that results are due to random variation. If they are due to random variation, then our results aren't valid. If they aren't due to random variation, then our results are valid. Not all forms of inquiry into nature are scientific. Scientific hypotheses have to be testable and have the potential of being rejected. Science depends on evidence that comes from reproducible and quantifiable experiments. And that is the end of your first video lecture on the nature of science.